Don't you just hate it when those insects buzz non-stop, disturbing your peace and focus? Annoying, right? But it would actually be more bearable if that's all they do. Because sometimes, their buzzing is accompanied by bites, which could be fatal if they carry the dengue virus or DENV, one of the world's deadliest and most prevalent viruses with approximately 400 million cases and 20,000 deaths per year. But how exactly does a small bite cause such a huge problem? Hi, we are JC Quintin, James Biñas, and Angelo Dela Cruz from the Philippine Science High School main campus, and we will be discussing our study on the in silico identification of dengue virus serotype DENV-3 RNA-dependent RNA polymerase inhibitor core structure.
After Dengue invades a host cell, its RNA gets translated by the ribosomes, which is cleaved into 3 structural proteins and 7 non-structural proteins. Among these, the non-structural protein 5, specifically its RNA-dependent RNA polymerase domain, functions on the replication of viral RNA by synthesizing multiple copies of DENV RNA through a template. Think of the RdRp enzyme as a power outlet by which the RNA template, which can be thought of as a plug, needs to connect to for replication to take place. So to prevent this, we would need to cover the outlet, and that's precisely what RdRp targeted inhibition studies aim to do. One of these studies identified HEE1/2 tyrosine as a potential inhibitor. However, despite the progress in this field of study, there are still no antiviral drugs against dengue to date.
Hence, our study aims to determine a core structure for a potential inhibitor of the RdRp of the DENV serotype 3 through the virtual screening of the National Cancer Institute Diversity Set 6, a chemical library which provides maximal structural diversity and pharmacologically favorable compounds. The outcome of the study may become a foundation for the next steps of the drug development process, like, for instance, a more focused high-throughput screening.
The study follows these major steps. First, the 3D conformers of the 1,584 compounds from the NCI Diversity Set 6 were prepared as ligands. Next, the DENV-3 RdRp crystal structure was prepared as a receptor by removing unnecessary molecules and adding polar hydrogens. Along with it, the grid box for molecular docking was prepared, taking into account the pertinent amino acid residues.
Then, we performed molecular docking using AutoDock Vina. Lastly, the ligands with binding affinities that were less than negative 9.5 kilocalories per mole and have either hydrogen bond or pi stacking interactions with tryptophan 795, which are based on how HEE1/2 tyrosine bound with the protein, were considered as top hits. We then identified common families among them, and their interactions with the protein were investigated.
As for the results, we identified 26 top hits from NCI Diversity Set 6, which were divided into structural families. Among them, sterines have the highest binding energies, ranging from negative 10.3 to negative 10.7 kilocalories per mole when binding to the RdRp.
In addition to their high binding energies, each sterane ligand showed similar interactions with the binding site of RdRp. Hydrophobic interactions with tyrosine 606 and tryptophan 795, hydrogen bonding with arginine 481, and pi stacking interactions with tryptophan 795. In case you're wondering how they have these common interactions, that is because of the similar structures of the sterane ligands. For example, besides the common interactions stated before, these two ligands, NSC60785 and NSC80997, have a more hydrophobic surface compared to the other one, NSC37641, and that allows them to form multiple hydrophobic interactions. Also, NSC80997 and NSC37641 have many polar groups around their structures relative to NSC60785, and this makes them interact with the binding pocket via multiple hydrogen bonds.
Due to their similar interactions, when the post pictures of the sterane ligands are overlaid, it can be seen that they are clumped together in the binding site. Their positions are near the priming loop, the part of RdRp that regulates the entrance and exit of compounds on the binding site. This suggests that the substrate would be blocked from binding with the binding site of RdRp, thus theoretically implying the inhibition of the replication of the viral RNA.
Therefore, we concluded that sterane may be used as the core structure of a potential DENV-3 RdRp inhibitor due to the high binding affinities exhibited by the sterane top hits on the positions they occupied within the binding pocket. This is significant because a newfound inhibitor core structure can pave the way for the development of future lead compounds that may eventually become drugs that can cure dengue. Maybe by then, all we have to worry about are the annoying buzzes. That is all for our presentation. Thank you very much for listening.